Mm. Hello, good morning, everyone. Am I audible?
Yes, sir.
Maybe, sir. Okay, all right. So I think, hello, sir. Namaskar. Professor Avinash Sonone, sir. Hello, Professor Avinash Sonone, sir. Am I audible? Sir, to the unmute. Unmute.
Namaskar.
Right. Hello, sir. Yes, sir. Namaskar, sir. Good morning, sir. Yeah. So actually, first of all, welcome you, sir, in this particular lecture series, which we had organized from 8th February to 20th February 2021 under the UGC-STRIDE program, as well as Maharashtra Academy of Sciences. So on the behalf of Shivaji University Kolhapur, Department of Biochemistry, Department of Microbiology, Shivaji University Kolhapur, we actually welcome you, Professor Avinash Sonone, sir, all the way from IIT Indore, Madhya Pradesh. Actually, before we begin, it is my kind pleasant duty to introduce Professor Avinash Sonone, sir. So, as we know, Professor Avinash Sonone, sir, is working in Department of Biosciences and Biomedical Engineering, Indian Institute of Technology, Indore. That is IIT Indore, Madhya Pradesh, India. So basically, his research work is particularly related to the disease biology and cellular immunological research that he is carrying out at the Indian Institute of Technology, Indore. So broadly, he is working in the area to understand the molecular mechanisms of Mycobacterium tuberculosis, tuberculosis pathogenesis in bone marrow stem cells and lung macrophages, host immune evasion mechanisms, development of active small drug compounds and antimicrobial peptides, and targeted drug delivery systems to improve the tuberculosis therapy. In addition, Sir is also working to develop novel asparaginase enzyme-based therapy for the treatment of primary and relapsed acute lymphocytic leukemia cells. So about his path in actually before joining to this IIT Indore. So let me first of all inform to all of our participants, dear students, and actually faculty members that actually Professor Avinash Sonone, sir, presently working with the same institute at IIT Indore as an associate professor from 2018 to 19. And then actually before that, 2015 to 18, she was working as a School of Biotechnology, KIIT University, Bhubaneswar, India. Then he was also working as a associate professor at the same institute, KIIT University, that is Bhubaneswar, India. Then actually he had completed several postdoc research, particularly at European Molecular Biology Laboratory, that is popularly known as EMBL, and all our students there you might heard about, or we know that EMBL, GenBank, and DDBJ, these are the gene sequence databases that we have learned, or we basically learned through our syllabus. So sir had completed his postdoc from 2007 to 2008 from EMBL, Heidelberg, Germany. Then before that, he had also worked as a postdoctoral fellow from 2006 to 7 at Max Planck Institute for Infection Biology, Berlin, Germany. And then actually again earlier to that, 2004 to 2006, he was in the Department of Medicine, Infectious Diseases, University of Florida, USA. So sir has got wide experience of research, teaching, and doing very active researcher in particularly important research areas on Mycobacterium tuberculosis, understanding mechanisms and development of new drug targets with respect to antimicrobial peptides. So actually, sir, welcome you all in this particular lecture series. Basically, we are very much thankful for you for accepting our invitation. Since last year, we are trying to invite you, and we are trying to organize your lecture for our student and faculty members. I am hope that this year we could. Actually, we are that much lucky enough to have you here on this particular platform. So, sir, before starting your lecture, I would like to inform you that we had organized this particular lecture series, and this lecture series had been inaugurated by our Honorable Vice Chancellor Professor D.T. Shirke sir, actually present Vice Chancellor, and the chief guest for this inaugural function was actually Professor Abdullah Shoyal from Jahangirnagar University, Bangladesh, with whom we had signed one MOU, and we are also doing active research in areas of microbiology and biochemistry as well as biotechnology. So there are several speakers that we had invited for this particular lecture series, and this is the basically actually third lecture which we are going to conduct. That is yours only. So, sir, again welcome you all. Welcome all students, participants, faculty members, and I request Professor Avinash Sonone sir to start his lecture. Thank you, sir. Hello. I think some issues are there with Professor Sonone sir. Hello, Professor Avinash sir.
I'm audible.
Yes, sir, you are audible, but your voice is breaking.
Audible?
Yes, sir, you're audible, but your video is not on. Maybe some internet connectivity issues there.
Is it?
Yes, sir. Now it is okay. Yeah, it is okay now, sir.
Okay. Yeah. So if I'm not audible, just let me know. There might be some
Sure.
activity issue.
We'll let you know, sir.
Yeah. So a very good morning to you all. First of all, I would like to thank organizers and particularly Professor Sonone for his kind invitation to accept this invitation for a couple of reasons. First, I really like to interact with, particularly with the college students and faculties, because I also studied in a very small place, near Malegaon.
Sir, your voice is breaking in between.
300, sorry. Is it okay now?
Hello, sir. Hello, sir. We cannot hear you properly.
I don't know.
Yeah, now it is okay, but sometime your voice breaks actually.
Okay. Is it okay?
Okay? Now it seems okay. Yes, sir. Now it's okay.
Okay. Yeah. So I really like to interact with the college students and faculties. So I was very this invitation and people.
Surrender Prasanga. Hello, sir. I think if you switch off your video, it would be fine actually.
What's
No, sir. No, we cannot see the slides. It is just white background is coming. Now, yes, sir. Now it is. We can see now.
you can see now, right?
Yes, yes. We can see it now, sir. Please go ahead.
Fine now.
Yes, it is fine now, sir. It is fine.
Okay. Okay.
So please
Yeah,
switch off your
sorry.
video and just keep only mic only so that network issue won't be there.
Okay. Yeah. So uh, some you know that today we are facing a huge problem of antimicrobial resistance. So if you talk about a particular country, for example, in USA, so some of you might have seen this data from the CDC. So more than 3 million drug registered bacteria are evolving every year. And there are more than 6 antimicrobial resistance. So how this antimicrobial resistance is evolving? Okay, so there are many drugs which were discovered a century ago. Now, this organism, period of time or through this evolution process, they have developed resistance against these drugs. And if you see the treatment of this multi-drug resistance bacteria now, or even these viruses, so earlier these infections, they were used to get treated effectively by using, I don't know, like 1 or 2 drugs. But now many of these multi-drug resistance infections, they require a combination of different drugs, and that involves like classification into first line drugs and second line drugs. So now what is first line and second line drugs? That these drugs, so first line drugs, these are the most effective drugs. Okay? So these are most commonly used for treatment of this infection. But what happens? These bacteria over the period of time, they develop different mechanisms. So we know that they act on the pathogens through different mechanisms. And there are some target sites present in these pathogens. But these pathogens, they modify these target sites either by introducing some mutations or by changing the structure of the target proteins. So through these different mechanisms, these pathogens, they develop this resistance. Now, resistance infections, they are treated
Hello,
by
sir,
a
excuse me.
combination of different.
Hello, sir. Excuse me. I think your slides are not moving, sir.
No. Actually, I'm still on the first slide.
Okay, sir. Okay, fine. Thank you. Okay.
Yeah.
Please go ahead.
Yeah. So this infection, just take example. Vaccine. Okay, sorry, this COVID vaccine. Use the combination of different drugs, which will have multiple target sites and which can kill this infection. So these drugs are now classified as a second generation drugs. So today I'm going to show you examples on the drug development which is happening in our lab, and we are working on development of both communicable as well as non-communicable diseases. So I will just give example of one drug now which is used for the treatment of the non-communicable diseases, for example, the blood cancer. And another example on this drug, which is used for treatment of the tuberculosis, which we know that is a communicable disease. So, okay, whether these slides are changing now? Yeah. So we know that there are many drugs which were discovered a century ago, and now, as I mentioned in my previous slide, these drugs, these microorganisms, they develop resistance against these drugs. So as you can see, the chronology of development or the discovery of different drugs. As you can see, when this drug was introduced, these drugs initially they were very effective in treatment of different infections, but after several years or decades, these organisms they develop resistance against these drugs. For example, the penicillin. Okay, so penicillin was very widely used to treat the gram-positive infections, but then after 10-15 years, many of these microorganisms they developed resistance against penicillin. There are similar other examples like tetracycline, erythromycin, methicillin. So these drugs, they were very effective in the beginning, but over the period of time, these microorganisms, they develop resistance against these drugs. So to overcome the challenges, now we need to come up with a very innovative concept that to make a hybrid molecules which will have multiple target sites and which can kill this drug resistance bacteria more effectively. So since this is the first interaction, I would just like to give you an overview about the research which is going on in our lab. So our lab mainly works on two diseases, that is tuberculosis and blood cancer. So in tuberculosis, as you know that this disease is caused by a bacteria called Mycobacterium tuberculosis. And this Mycobacterium tuberculosis, it has many different types of proteins. And one of this important class of protein which is present in Mycobacteria is called as the glycoproteins and glycolipids. Okay. And these proteins, they play an important role in the pathogenesis of microorganisms. So these proteins, they help this pathogen to avoid recognition by our host immune response. These drugs, they provide resistance against the several drugs. These drugs, they provide this bacteria to survive in macrophages or in lung, this microenvironment. So we're trying to understand what is the role of this Mycobacterium tuberculosis glycoproteins and glycolipids in pathogenicity. It means how these proteins, they help in the disease progression. These proteins are also very immunogenic in nature, and these proteins can also be used as a vaccine candidate. Okay? Because when these proteins are exposed to macrophages and T cells, these proteins were found to induce or activate these T cells or even B cells to produce the antibodies. So we have some data which indicates that these proteins can also be used as a vaccine candidate. And if you look at the structure of Mycobacterium tuberculosis, it has very complex. The 7% content, the drugs, they are not able to penetrate into the cell wall of this Mycobacterium tuberculosis. And this causes resistance to the drugs. So we're trying to understand that how these proteins, they play a role in development of this drug resistance against several anti-TB drugs. Okay. Another area we are working on, what are the different changes are happening in the DNA when our cells get infected with Mycobacterium tuberculosis, and these changes are called as the epigenetic changes. Okay. We know that this tuberculosis, it mainly occurs, the primary site of infection is lungs. Okay, so when this Mycobacterium tuberculosis enters in our body, it first gets deposited into the lungs, and where we have these lung macrophages, or which are also known as alveolar macrophages. So these alveolar macrophages get infected with this Mycobacterium tuberculosis. Okay? And these alveolar macrophages, this Mycobacterium tuberculosis, it manipulates the function of these alveolar macrophages so that these our lung macrophages, they are not able to kill this Mycobacterium tuberculosis. And as a result, this bacteria, it survives for a longer time. It causes chronic tuberculosis. And because of this chronic infection in the lungs, that also leads to lung cancer. Okay. So we're trying to understand what is the correlation of this chronic tuberculosis with the lung cancer development? Okay. Another area we are working on is on the bone marrow stem cells, because uh, earlier it was thought that the tuberculosis mainly happens in the lungs. But what happens when our immune system is not able to recognize these pathogens in the lungs? These pathogens, the Mycobacterium tuberculosis, it survives for longer time, and then it goes into the bone marrow. Okay, so bone marrow means you might have seen in the bones, inside the bone, there is a flesh like the spongy flesh. Okay, that is called as the bone marrow. And this Mycobacterium tuberculosis, it goes in that spongy or in the bone marrow, and it lives there for several years. Okay, and this is a very protective environment for any pathogen, because the drugs, they are not able to penetrate easily into the bones. Okay, and as a result, these drugs, they are not able to kill the microorganisms which remain inside the bone marrow. Okay, so this TB remains in the bone marrow, and we're trying to see that how this tuberculosis, it modulates the function of the bone marrow and helps this pathogen to survive for a longer time. Another area we're working on is development of new anti-TB drugs, which I'm going to discuss in next slide. Um, so another area we're working on is on blood cancer. So okay, so this blood cancer is mainly treated by a combination of these different drugs, and one of the molecule which is used in blood cancer treatment is the asparaginase. We're trying to develop the asparaginase-based therapy to improve the treatment of primary as well as relapse this blood cancer. And since it has been found that the chemical modification of these therapeutic molecules, it increases the efficacy of these molecules to kill the cancerous cells. So we're trying to introduce different types of chemical modifications in this enzyme so that we can increase the therapeutic efficacy or anti-cancer efficacy of these asparaginase drugs. So these are the different areas we are working on. And this topic is more focused on drug development. I will just give you a couple of examples. The work which is happening in our lab. So our lab is mainly working on development of anti-tuberculosis drugs, because as you may know that now there are multiple drug resistant and extremely drug resistant tuberculosis strains are present. So what is multiple drug resistant tuberculosis? Because if you know the different types of antibiotics or drugs which are used for the tuberculosis treatment, these drugs are classified into two these classes. That is first line drugs, which involves isoniazid, rifampicin, then pyrazinamide or ethambutol. So these are the first line drugs which are used for treatment of tuberculosis. And then there are second line drugs, because when these drugs, first line drugs, they are not able to kill tuberculosis, Mycobacterium tuberculosis, then the combination of first and second line drugs are used. But now there are several reports which have shown that this Mycobacterium tuberculosis is even has developed resistance against the first line and second line drugs. So our lab is working on development of new class of antibiotics which can treat the TB infections which are resistant to first line and second line drugs. As I mentioned, we are also developing the asparaginase-based therapy for treatment of blood cancer. And since this COVID pandemic has started, so our lab also started to work on the development of prototypes, which will help in killing of these COVID viruses which are present in the air. So we are developing a prototype through which the infected air, particularly in big hospitals or at crowded places, these viruses, they get expelled into the air. Okay. So we are developing a prototype through which this contaminated air will pass through and it will kill the viruses which are present in the air. So these are the three major areas our lab is working on in terms of the drug development. Now coming back to the drug, which our lab is primarily working on, is the development of the M-aspart drug. So this is the name which we have given after discovery of this drug in our lab. And this M-aspart drug, is mainly used for treatment of primary and relapse acute lymphatic leukemia, which is a type of blood cancer. And this molecule is under now clinical trial. So I will just show you some of this data or the development journey of this drug. So before I start, probably you know that there are several stages of blood cancer chemotherapy. And these phases are classified as induction phase, consolidation phase, and maintenance. Now, what is induction phase? When the blood cancer is developed, so in the initial few phases, uh, these leukemic cells or cancerous cells, they multiply very rapidly. So, uh, in this phase, the induction phase, the different combination of these drugs are used, like anthracycline, steroids, vincristine, then methoxytricate. Okay, so these are the different drugs which are used, and these drugs they kill these rapidly dividing cancerous cells. Okay, and this is called as, so this is a very rigorous phase of treatment. And this phase is called as the induction phase. So in the induction phase, the rapidly dividing cancerous cells are killed by using a combination of different drugs and which involves asparaginase as a main component. Okay. So once these rapidly dividing cells are killed, still there are some residual cancerous cells remain. Okay. So these residual cancerous cells, they are killed by uh, another combination of these different drugs, and this phase is called as the consolidation phase. In consolidation phase, the residual cancerous cells are killed by using a combination of different drugs as shown here. And this combination of different drugs also involves asparaginase as an important component. And then the last phase is the maintenance phase. So when these residual cells are killed, then the doses of these chemotherapy drugs is reduced and it is given for several weeks just to maintain or not allowing these any new cancerous cells to develop. So that is called as the maintenance phase. So if you look at to the different stages of blood cancer chemotherapy, you can see that the asparaginase drug is used at in every stage or in every phase. So this is a very important chemotherapeutic drug used in the treatment of blood cancer. So there are various studies which have shown that when this L-asparaginase, which is basically produced from the E. coli strain, and when this L-asparaginase drug is used in chemotherapy, then the survival rate of these patients, it increases to more than 80%. And there is no substitute for L-asparaginase. So this drug is used in treatment of this blood cancer. So there are various types of asparaginase drugs are available in the market. I have just shown few examples here. For example, Elspar, Oncaspar, Rylaze, Nunez. So these are the different drugs. These are basically the L-asparaginase formulations, which are marketed by different companies by different names, which are available in the market today. However, these asparaginase drugs, it has several limitations. Just discuss about that. So this asparaginase is an enzyme. Okay. So the current asparaginase drug, it has very low enzymatic activity. Okay. Because of this low enzymatic activity, the patient they need to take repeated doses of asparaginase. Okay. And because of this repeated doses, it causes this toxic effect to various organs. Okay, like to the spleen, kidney, liver, and these repeated doses also causes the allergic or hypersensitivity reactions in the patients, as you can see here. Okay, so these are the allergic or hypersensitivity reactions which happens due to the administration of repeated doses of asparaginase. The another major problem associated with this asparaginase drug is that it is highly neurotoxic. Okay, when it is given to these blood cancer patients, this repeated doses, it causes toxicity to the brain. And this brain toxicity, then it leads to various neurological disorders. Okay. Another major problem with this drug is that it is highly immunogenic in nature. Now, probably you know that what is immunogenicity. Right? So, immunogenicity it is the ability of the protein to produce the antibodies in host body. Okay. So this asparaginase, as I have shown in the previous slide, that it is a bacterial protein. It is derived from the E. coli. And when this bacterial protein is given to human as a drug, the human body, it recognizes this protein as a foreign protein. Okay? And because of its foreign nature, our immune system, it starts to react against this foreign protein and it produces antibodies against this asparaginase. Okay. And when these antibodies are produced, these antibodies, they bind to this protein or to this asparaginase drug and it neutralizes this protein. And when this drug is neutralized, what will happen? This drug will not be able to kill the leukemic cells. Okay? And because of this inactivation, uh, there is a huge problem in the treatment of relapse blood cancer. Relapse means when the cancer is caused, when it is treated, then it is treated, is cured, and then after we see that after a few years or after a few months, again we see a recurrence of the disease. So that is called as the relapse cancer. So because of this presence of these antibodies, when there is a relapse, when this drug is given second time, asparaginase drug, so the antibodies which are present in the patient body, these antibodies, they go and bind to this drug and it neutralizes. So the treatment of relapse blood cancer is a big challenge. Okay. And because of these properties, this asparaginase drug, it causes allergic reaction, uh, because of these repeated doses of the high doses, it causes toxicity to liver, spleen, lymph nodes, and various other organs. So now we need to develop a new drug. And this, since I thought that there might be some students might be attending this talk, so I just put this quote specifically just to show that someone is sitting in the shade today because someone planted a tree. So just take example of COVID virus. Okay. So when this virus was detected first time in 2020, so nothing was known about it. Then the whole entire scientific community across the globe, they started to work on it, and they in like record time we got the vaccine approved within. I mean that is the history in the medical science, that the drug came into the market after 1.5 years. Okay. That too, like several drugs. So, so now, as we might have seen in the third wave, okay. So even though the infectivity rate was very high, but the mortality rate is significantly less. It is mainly because whatever discoveries were happened in last 1 year, like vaccines or different drugs, these vaccination, it gave protection to a lot of people, and that's why we see although the infection rate is very high, but the mortality rate is very less. So as a student, as a young generation, now you have to think about like very innovative ideas out of box, how we can do science which will be helpful to the society and it will help the coming generation in overcoming the challenges which we are facing today. So probably you know the process of hematopoiesis. It is a process which is happening in the bone marrow where production of different cells takes place. So I will not go into the detail. So just give you overview about the blood cancer. So normally what happens when these cells are produced in the bone marrow? Okay, as white blood cells, involves your neutrophils, macrophages, WBCs, different types of cells. So these cells, they perform a different immune functions. These cells get converted, particularly the white blood cells, okay, when they become leukemic cells or the malignant cells, we know that these cancerous cells, their doubling time is very short. So they multiply very rapidly. So what happens when these cancerous cells are produced in the bone marrow? Because of its high multiplication rate, over the period of time, the number of these malignant cells or leukemic cells increases, and the number of the healthy cells or non-malignant cells, it decreases. Okay, so these malignant cells they are not able to perform the immune function, okay? Because we know that these white blood cells, they are mainly responsible for to kill the infectious agents. Okay, whereas these malignant cells, they are not able, they lose their affinity with this property to kill the infectious agents. So over the period of time, these malignant cells, they their number increases, and that leads to a blood cancer. Okay, and there are different types of leukemia, like AML, CML, then ALL, and among these different types of leukemia, this acute lymphatic leukemia, it is a major type of blood cancer. About 70 to 80% of the blood cancer are diagnosed with the acute lymphatic leukemia. And this disease is also very common in children below 15 years of age. So about 23% of the all cancers which are diagnosed in the children, it represents or it has the acute lymphatic leukemia. And if you look at the number of cases diagnosed in India, so every year more than 35,000 new acute lymphatic leukemia cases are diagnosed in India. And globally, there are more than 900,000 ALL cases are found. But as I mentioned, the asparaginase, it is a very crucial component for the treatment of the leukemia, and there is no substitute for the asparaginase drug. Now, how this drug it kills the leukemic cells? As I mentioned, this asparaginase is an enzyme which converts this asparagine into a aspartate amino acid. So in healthy these individuals, what happens? This aspartate and asparagine, these both amino acids are present in our blood. So the healthy cells, it can be your macrophages, neutrophils, eosinophils, dendritic cells, all sorts of cells. Okay. So these cells, they take up this aspartate from the blood. Okay. So this aspartate goes inside the blood, inside the cells, and these cells, they express an enzyme called as asparagine synthetase. And this enzyme, it converts this aspartate into asparagine. Okay. And this asparagine amino acid is used for the protein synthesis by the cells. And then when this protein synthesis takes place, the cells will multiply and they will survive. Okay. So this is how this asparagine is very important for the protein synthesis and the survival of the cells. The another mechanism is that this asparagine is also present in the blood. So this asparagine is directly taken up by these healthy cells and use this asparagine for the protein synthesis. And as a result, the cells will multiply and survive. But what happens when these healthy cells get converted into the cancerous cells? So these cancerous cells, they do not express this asparagine synthetase enzyme. So in this case, what happens? The aspartate which is present in the blood, it is taken up by these leukemic cells or these cancerous cells. But because of this absence of this asparagine synthetase, this aspartate is not converted into asparagine. And since we know that this asparagine is important for the protein synthesis, but because of this lack of this enzyme, this aspartate is not converted into asparagine, and it affects the protein synthesis in the leukemic cells. Okay, so these cells, then they are dependent on the asparagine which is present in the blood. Okay, because there is no conversion of aspartate into asparagine due to absence of this enzyme in cancerous cells. So these cells, they take up this asparagine which is present in the blood and use this asparagine for the protein synthesis and for the survival of the cells. So what happens when this L-asparaginase is given as a drug to these cancerous cells? So as we have seen that there is no, these cells, cancerous cells does not express the asparagine synthetase. So there is no conversion of aspartate into asparagine. So these cells take this asparagine from the blood and use this asparagine for the protein synthesis. When this drug is given to these cancerous patients, this asparagine, it converts the asparagine which is present in the blood into aspartate. Okay, so basically this administration of this asparaginase, it depletes the asparagine which is present in the blood, and as a result, these leukemic cells or the cancerous cells does not have asparagine for the protein synthesis. So these cells, they undergo apoptosis and multiply. So this is how this asparaginase, it acts as drug to treat the cancer patients. The another drawback of this drug is that since this is a bacterial protein, it is immunogenic in nature, and when it is given to human or to the this ALL patients, So when this asparagine drug is given to the ALL patients, this ALL patient, they recognize this protein as a foreign protein and it produces antibodies against this asparaginase drug. Okay? And when there is relapse of cancer, when this drug is given again, so these patients, they already have taken this asparaginase therapy before. Okay, so this ALL patient, they already have antibodies against asparaginase. When this drug is given, the antibodies which are already present in the relapsed ALL patients, these antibodies, they bind to this asparaginase drug and neutralizes it. And because of its neutralization, this enzyme is not able to kill the leukemic cells. So if you look at to the national status of this acute lymphatic leukemia, so this medical data has shown that about 70% of the ALL patients in India, they come from very poor this background, cannot afford the treatment because the blood cancer treatment, it may last for 28 to 30 weeks. Okay, so the treatment duration is very high, and these poor people, they are not able to afford this treatment cost. Whereas only 25%, 25% it comes from the middle class, and only 5% it comes from this rich families which can afford this treatment. So because of this socio-economic status, these many people, they are not able to afford this good quality of this asparaginase, and therefore the outcome of this blood cancer therapy is relatively very less effective as compared to the developed countries, because the very good drugs, asparaginase drugs, which has this less side effects and more efficacy, the total treatment cost, it costs around $140,000, okay, for 23 weeks, and which is quite expensive. So the most of the people in developing countries, they are not able to. So then how to overcome this problem? So our lab has been working on that, how to, okay, now there are several asparaginase drugs are available, but then overcome the limitations of the side effects which are seen in the asparaginase drug. So our lab is developing a new asparaginase drug which will produce less hypersensitivity or allergic reactions. Okay. Uh, this Caspar or it is PEG asparaginase drug, it is the most commonly used, but the treatment cost is between $70,000 to $160,000. So the treatment cost is very high, and this cannot be afforded by the middle class or the poor class family. And as I shown before, that this is the major type of cancer identified in blood. So we need to develop a product or new asparaginase drug which will reduce the treatment cost, which will not induce any side effects, and which will also improve the treatment of relapse this blood cancer. So this is the structure of asparaginase. I will not go into the detail. So our lab, we use a protein engineering approach. So what is protein engineering? So we modify the properties of this asparaginase drug by modifying the B cell epitopes and various amino acids which are present in this protein, which are responsible for these side effects. So through protein engineering, we modified this B cell epitopes and several amino acids which are present in this drug. So through this protein engineering, we developed around 12,000 new mutants, and each mutant had mutations at various positions. Okay? And we made mutations. First, we identified the amino acids which are responsible for the neurotoxicity, or the amino acids which are responsible for the immunogenicity, the amino acids which are responsible for the stability. So we mutated these amino acids through this protein engineering approach and developed around 12,000 mutants. And then we screened all these mutants and tested each one of these mutants for the properties like we check their glutaminase activity, whether any mutations cause reduction in the glutaminase activity. We also check whether these mutations, they increase the enzyme activity, whether any mutations, they reduce the immunogenicity. So we screened all these 12,000 mutants And then finally, we found that there are 4 mutants which had mutations at different positions. These mutants, they showed these desired properties, such as these mutations, they increase their enzyme activity. We found that some mutations, they reduce the glutaminase activity and also immunogenicity. So these mutations were present in different mutants. So then what we did, we combined all these mutations in and we named this enzyme as the M-aspar enzyme. And this M-aspar drug, now this is a single enzyme which has different mutations in it. And this enzyme has high enzymatic activity, low glutaminase activity, and also reduced immunogenicity. So I will just show you some data with this enzyme. Mainly, I'll just show this the data which we had derived from cancer patients and the mice. So as you can see, we check this neurotoxicity. So this mutancy means this is MSFR. Okay. So there is significantly less glutaminase activity, which causes less neurotoxicity. Um, as I showed, that this is a immunogenic protein, and when it is given to patients, it produces antibodies. So we did an immunization study where we took these VALPs mice, and these mice were injected with this EMASPA drug on day 1. Then after 21 days, we gave second dose of this EMASPA. Then after 45 days, we gave third dose. And then after 66 days, we gave fourth dose of this EMASPA. And then after 75 days, we collected this serum from this immunized mice, and then we checked production of antibodies in the mice. As you can see, the mice which were given the native form of this asparaginase, there were more IgG and IgM antibody production. Whereas the mice which received this M-aspar drug, there was significantly less production of IgG and IgM antibody. It indicates that this enzyme or this M-aspar drug is significantly less immunogenic, or it does not induce the production. Then, as I mentioned that this asparaginase drug also binds to the antibodies which are present in the cancer patients. So we studied whether our M-aspar drug binds to antibodies which are present in the cancer patients or not. So what we did, we recruited the three classes of patients. The patient A means these patients they are diagnosed with the blood cancer, but they have not taken any asparaginase drug. Okay, so if they are not taken any asparaginase drug, means there is no asparaginase antibody produced in patient A. Okay, in patient B, these patients they are diagnosed with the blood cancer, and this is started to take asparaginase therapy 3, 4, 5 days before. So since the duration is very short, there will be few antibodies will be produced in ALL patient B class. Whereas in ALL patient C, these patients, they will taking this asparaginase therapy for more than 45 days. So it means these patients are expected to have more anti-asparaginase produced in their body. So we took this serum from this patient A, B, and C because we know that these antibodies are mainly present in the serum. And then we tested whether these serum antibodies from patient A, B, and C, they bind to this. As you can see in mutant C, these antibodies which are present in primary and relapsed ALL patients, it binds significantly less to our M-ASPAR or to the mutant C as compared to the native enzyme. So it shows that the M-ASPAR drug does not bind to the antibodies which are present in the patients. Therefore, this enzyme can also be used for the treatment of the relapsed blood cancer. Then we did pharmacokinetic studies. We checked when this drug is given to mice, how long this drug remains in the blood. So what is the drug concentration? What is the half-life of this drug in the blood? How fast this drug is eliminated from the mice. So these all studies are called as the pharmacokinetic. Pharmacokinetic, what we did, we took this biopsy mice. Then one set of mice was given this wild type asparaginase. Another set of mice were injected with this M-aspar, our drug. And third control means these mice did not receive any treatment. And then after injection, we collected this blood after 5 minutes, 30 minutes, up to 24 hours. And then we checked these parameters, the concentration, half-life, and elimination rate. So as you can see that as compared to the wild type, and also we use another leucogenous asparginase drug as a control. So as you can see, after 24 hours, this M-aspar over drug, the concentration in the blood was significantly higher if you compare. And also its half-life was significantly more. So if half-life is more, it means this drug remains in the body for longer time. And since the exposure time is more, it will be able to kill these cancerous cells more effectively. Okay. And elimination rate was less, okay, as compared to the wild type. It means this drug is, M-aspar drug is eliminated from the body very late. Okay. So because of this less elimination, the concentration will remain in the body for longer time. So these are all desired properties. And this is a very important experiment. So here what we did, we developed this blood cancer in mice. So what we did, we took the cancerous cells, blood cancer cells, and we injected these 10 million cancerous cells into Mars-kid mice. We allowed these cancerous cells to develop, to multiply in the mice. This is called as the xenograft mice model. So when this cancerous cell population reached to 5%, okay, of the total cells, then these mice were treated with the asparaginase drug, that is our M-aspar drug, and the wild type. And then 2 more injections were given after 7 days. Then after 14 days, we sacrificed these mice and then we checked that how many cancerous cells are present in these mice. Okay, so what we observed, that when these mice were injected with our drug, more than 90% of the cancerous cells were killed when these mice were given this M-aspar drug. Whereas when these mice were given this wild type asparaginase, then only 30% of the cancerous cells were eliminated. Okay, so it is 90% versus 30%. Okay, when we check the survival of these mice, we found that when these mice were given with this our M-aspar drug, only one this mice died. Okay, so out of the 10 mice, 9 mice they survived. Whereas in case of this wild type, all mice were dead after 20 days. Okay, so it means this drug it is able to kill 90% of the cancerous cells which are present in the mice, and it also increases survival of these mice. So these are all desired properties. And then finally, we also check whether this drug has any toxic effect on different organs like spleen, liver, kidney, brain, whether it changes different blood parameters or not. So we did this by single dose and multiple dose studies. In single dose, these mice were given the single dose of the asparaginase and M-aspar drug, and in 3 doses, because in cancer, the patient needs to take multiple doses of these drugs. Mimic that, we also did this through 3 dose level studies where these mice were given this M-aspar drug or the wild type drug at 3 days interval. Okay, so 10 days, we give 3 doses. And then we sacrificed these mice and studied the histopathology of liver, kidney, spleen, various organs. I'm not going to show that data, but we show that it does not cause any toxicity to the mice. So now finally we have the M-aspar drug, which we found that is highly stable in the blood. It does not produce any antibodies in the mice, so there won't be any allergic reactions. Our drug does not bind to the antibodies which are already present in the ALL patients, so this can improve the treatment of relapsed ALL. I have shown that our drug also has better pharmacokinetic properties, and it is also able to kill 90% of the cancer cells. And since this drug does not involve any chemical modification, so it can reduce the treatment. Now, this drug, we have signed agreement with Tata Memorial Hospital, Cancer Hospital in Mumbai, and a pharma company in Mumbai. So we are doing this GMP manufacturing of this drug in this pharma company, and very soon we are going to start clinical trial of this drug in Tata Cancer Hospital, where we'll select at least 30 to 40 cancer patients and we'll give our drug to these cancer patients, and we'll see whether it improves the cure of the blood cancer. So we believe that this muscular drug will have reduced side effects. It will be highly stable in serum. It can also improve the treatment of cancer, and it can be cost effective because it does not involve any chemical this modification. So this development of this drug was widely covered in several media across India. Also, it was covered in various news channels. For this, we also very recently we received a National Bioentrepreneurship Award for developing this new drug, and also a technology development award. But still, that is not the end. That's why I want to tell, because you see, sometimes our cancer cells may also develop some mechanisms to reduce the efficacy of this drug. So to think in future, so we have good molecule, but we are trying to improve the therapeutic efficacy of our drug. So we are now started to work on third generation of this asparaginase, where because when this drug is given to the patients, it goes in the blood, and in the blood there are various these molecules are proteolytic enzymes are present in the drug in the blood, and these proteolytic enzymes will degrade this asparaginase drug because it is a protein. Okay, so now we are trying to do the chemical modification of this enzyme and trying to develop the third generation. So this chemical modification will improve the stability of this drug in the blood so that the patient need not to take the repeated doses. That will reduce the treatment cost. And since the doses are less, it will also not cause any toxic effect on liver, kidney, brain. Okay. So this is the work currently undergoing in our lab on development of third generation. So I will not talk about the TB now, since uh, maybe at some other platform I will discuss the development of the anti-tuberculosis drug, if you wish. So maybe we can just have an answer. So just to show you very briefly, this tuberculosis. Now there are these multiple drug resistant and extremely drug resistant tuberculosis, and there are different drugs are already used. These are called as first line, second line, and third line drugs for TB treatment. But now these multiple drug resistant and extremely drug resistant strains, they have developed resistance against these drugs. So now we need to develop new class of drugs which will effectively kill this multi-drug resistance bacteria. So our lab is working on development of some novel benzofuse heterocycles, which can improve the treatment of blood cancer. And we also got the Indian and US patent now for this drug. So we have developed one benzofuran, this drug, which inhibits. So this is the cell wall of Mycobacterium tuberculosis. Okay, this cell wall is mainly responsible for resistance to the drug. So we have developed a drug which blocks the synthesis of one of the enzyme which is important for the cell wall synthesis. As a result, the cell wall synthesis of this Mycobacterium tuberculosis is hampered, and it helps in killing off this drug-resistant bacteria. So we did several computational studies. Now we have filed a US patent. So this is the molecule which we have developed, and this molecule was found to be very effective against the drug-resistant tuberculosis. And we have proved this by using a zebrafish model, where we deleted the target protein of this drug, and then we infected this fish with the Mycobacterium strain, which is where the expression of this target protein is not there. So this is a mutant strain. So we infected this fish with the wild type Mycobacterium, then the Mycobacterium where this protein is or this gene is deleted. And as you can see, when these fish were infected with wild type marinum, it develops a tuberculosis, TB. Okay, and when this gene was deleted, then there was reduction in the TB development or the symptoms of TB. Now we are developing a drug which will inhibit the synthesis of this protein. So we found that this protein is mainly responsible for the nucleotide biosynthesis. This drug, it will block the nucleotide synthesis in Mycobacterium tuberculosis. As a result, the bacteria will. So finally, I would like to acknowledge the funding received from various funding sources, BIRAC, Bioignition grant. Recently, we got early translational accelerator grant from BIRAC to start these clinical trials. And we also acknowledge the support received from SERS, DST, ICMR, BRNS. Of course, I mean, this work was done in collaboration with various other groups at Tata Hospital, this ACTREC, and the various collaborators from different, yeah, and so thank you very much for your attention, and now I'll be happy to.
Thank you so much, sir, for giving us a valuable information about development of next generation drug in treatment of tuberculosis and blood cancer, that is acute lymphoblastic leukemia. You have also given us a valuable information about EMASPA drug, different phases of treatment of this drug and chemotherapy. You have also given us the information about combination of this drug, limitations, properties, nature of this drug, structure, effects, and function of this drug. Thank you so much, sir, for sharing with us your experimental study of drug on mice. It will definitely very helpful for us in future, sir. Thank you so much.
Thank you very much.
Just a minute, Tripti, I think invite Shailesh sir to give propose vote of thanks.
Yes, sir. Before that, if any student have any question, they are free to ask here.
Hello students, it is time for question answers. If you have any questions, any doubts, please raise your doubts. I think no any questions. Tripti, go ahead.
Yes, sir. Now I request Dr. Wagmare sir to give vote of thanks. Over to you, sir.
Okay, am I audible?
Yes, sir.
Okay, respected Professor K.D. Sonone sir, Head Department of Microbiology, Shivaji University, Kolhapur. Today's eminent speaker, Professor Avinash Sonone sir, Department of Biosciences and Biomedical Engineering from IIT Indore. All the faculty members, my dear colleagues, and the dear students. As we know that this is the third session online lecture on the whatever the online lecture series we organized under the UGC-STRIDE program with the collaboration of Maharashtra Academy of Science Kolhapur chapter on the advances in life science researches. We have gone through that lecture which is delivered by the Professor Avinash Sonone sir, and I think this would be the very good lecture with respect to the whatever the online lecture series we have organized. That is nothing but advances in life science research. Because if you have seen that lecture, in that we have gone through the microbiology, we have gone through the biochemistry, we have gone through the biotechnology. In that, sir has very well explained about what is the leukemia. In that leukemia, what is the role of aspergillus? Again, that aspergillus, how it is responsible for that blood cancer. And if you want to treat that, what would be the role of aspergillus in that? So I think this would be the very good lecture with respect to our online lecture series, and this will very much helpful for our students to get in the future to get the ideal the pipeline for the research. So on behalf of Department of Microbiology and Biochemistry and the Shivaji University Kolhapur, I am very much thankful to the Professor Avinash Sonone sir for his valuable time and accepting our invitation and giving such a wonderful lecture on the topic of development of next generation drugs. I'm also thankful to the Professor Sonone sir for attending this lecture. I'm also thankful to the all faculty members and the research students and the dear students for attending this online lecture series. Thank you, one and all.
Okay, yeah. Thank you very much.
Hello, sir. Thank you very much.
Yeah,
Professor
thanks.
Sonone, sir. Hello, sir. Could you please switch on your video so that
Yes,
you
yes.
can have one snap?
Yes.
Yes, sir. Please.
I hope it was audible, right? Later on was.
Yes, sir. It was audible. It was very nice, and it was very wonderful lecture, sir.